Welcome everybody to the last Drug Hunter Flash Talk of the year 2025. We're very excited to be here today, and we have a special for you. So it's going to be a flash talk by myself about the molecules of the year in 2024. And we're going to dive right into the presentation of the molecules of the year in 2024 and the scientific highlights of these. And how we will be spending the next about 50 minutes. I think the presentation will be about 40 to 50 minutes, and we'll make sure we have 10 minutes for questions. So we'll first, for those of you who are not so familiar with Drug Hunter, we'll first do a little bit of an introduction into what Drug Hunter is. We will share the past molecules of the year and talk a little bit about the process. And then I'll dive into the meat of the presentation, the scientific highlights of the 2024 Molecule of the Year candidates. And ultimately, I'd encourage you and everybody here in the audience to submit molecules that you think are worthy of nomination for the 2025 Molecule of the Year candidates. And we'll have that vote in the next year. So that'll be super exciting. So let's get started. What is Drug Hunter? What are we trying to do? So Drug Hunter is drug discovery distilled. So the problem that we're trying to solve is that many scientists have in the industry. There's so much information out there in many peer-reviewed academic publications, and there's so many journals. There's conferences going on with posters and presentations, and you have patents, you have regulatory filings, financial filings, and within this mountain of information, it's really difficult for a single scientist to get to the knowledge, to get to that actionable knowledge that you can apply on your program. And we're trying to do that. We're trying to monitor all these different sources and distill the knowledge into case studies, into target reviews, modality reviews, technology reviews, so that you have the most important knowledge at your fingertips that you can then search on a single knowledge platform that is Drug Hunter. And what differentiates us from legacy providers is we're mostly really focused on the science. So we're trying to talk about, why is the industry interested in a particular target, or why did, the industry discontinue clinical trials in a particular target area? What are some challenges in maybe hit finding? What are new technologies that are applied, and what are benefits and challenges of these new technologies? What are some, problems that you may have in lead optimization? How have teams previously overcome those problems? May that be efficacy or toxicity problems, and those kinds of things we will analyze for you and make those available on our platform for you to be able to search those. We're proudly serving over 200 research organizations worldwide. That goes from large pharma organizations to biotechs to even investors and nonprofit organizations as well. And I'd encourage everybody in the audience to try out some of our new tools that we've launched in the past year. So we've dramatically improved our patent search. So you'll be able to actually search by target, by company, and you'll get a brief annotation every week. We're actually diving into the patent literature, distilling out those important information from the patents for you so you don't have to browse that whole document. And you're seeing trends so much easier. And on top of that, you have the monthly patent highlights as well, where we're doing a deep dive into the data from the top 5 to 10 patents every month. Also, our platform is completely structure searchable, so you'll be able to search through FDA-approved drugs, modern clinical candidates, and Drug Hunter molecules as well. And recently, we opened a disclosure platform. So as I pointed out in the beginning, there's so many disclosures happening throughout the literature and at conferences. And in order to monitor those, you can find those in the disclosure database with their structure, with their target, and also with an annotation from our scientific content team as well. So I'd encourage you to try out those new features as well. And Drug Hunter is really made by industry scientists for industry scientists. Our team has combined more than 100 years of drug discovery experience. We've been working in large pharma organizations such as Amgen, Pfizer, Novartis, and Lilly, and many, many others, as well as in biotechs really across the world, from San Francisco in the Bay Area, where I am, to San Diego, Boston, Basel, and even in the UK as well. We're proud to have a stellar, in my opinion, scientific advisory board that helps us with like directions and understanding what key opinion leaders in the industry are interested in, what are the challenges, what are some of the problems that the industry is trying to address, and how can Drug Hunter help address those problems. And with that, I'd love to dive into some of the past molecules of the year and talk first a little bit about the molecule of the year process. So in order to nominate a candidate for the molecule of the year process, the structure of the molecule needs to be disclosed. So we're not taking guesses, essentially, at the structure. In addition to that, there also needs to be like a catalyst event that happened this year that may be a first-time disclosure, or that may be a regulatory filing, or even like a clinical update on a particular compound. And then in terms of nominations, everybody actively working in R&D can nominate any molecules, even molecules that they have worked on and that their organization developed themselves. But in the voting process, we actually scrape votes that are on molecules that the organization of the voter is developing. So there's no self-votes allowed. There's no popularity contest. But very oftentimes, the industry, agrees that one molecule was really the standout in a particular year. I mean, it's very difficult to choose. Frankly, it's like choosing between your favorite children. But largely, we often come to an agreement. And with that, what were the molecules of the year in the past year? So the first molecule of the year ever at Drug Hunter in 2020 was lenacapavir, HIV capsid assembly modulator. In my opinion, a game-changing molecule that has changed the treatment landscape for HIV. And it's been also the science breakthrough of the year in 2024. So one of my jokes is, if you'd like to see breakthroughs 4 years after Drug Hunter, you should go read it Science. But of course, that's a joke. So the second one is nirmatrelvir in 2021. And nirmatrelvir is, of course, Pfizer's reversibly covalent Mpro inhibitor that helped us out of the COVID pandemic. And I think this molecule taught the industry a very important lesson of how can we accelerate drug discovery programs in general. And I think that has given many leaders food for thought as of how to do that. In 2022, our molecule of the year was deucravacitinib, a very interesting kinase inhibitor, TYK2 inhibitor that was invented by BMS and was actually discovered in a phenotypic screen. And it's really notable because it's a kinase inhibitor in oncology in immunology that you don't see that often. Oftentimes you see kinase inhibitors in oncology, and was also discovered by a phenotypic screening. Typically, kinase inhibitors, you would think about structure-based drug design and identifying those in a different manner. And also, deucravacitinib was the first de novo deuterated drug, meaning that the hydrogen version of deucravacitinib was not actually approved, and the deuteration is important for the half-life and the low dose of deucravacitinib, as well as for the selectivity of the compound at TYK2 over other JAK kinases and the entire kinome as well. And then in 2023, the molecule of the year was enlicitide, this wonderful macrocyclic peptide PPI inhibitor, PCSK9 inhibitor from Merck. And I think this is a whole new molecule class. It's comparably small for an antibody, but comparably large for a small molecule drug. And yeah, inhibits basically the interaction between the LDL receptor and PCSK9. Again, it's an orally available drug, which is very, very challenging in this biologics-dominated space. So Repatha is a clinically approved antibody that targets PCSK9. And now enlicitide enters this field as an oral option for hypercholesterolemia. And I think that's just a really remarkable entry. The design of the molecule was really very, very remarkable. And with that, we will dive into the molecule of the year candidates for 2024. I'll talk a little bit about laroprostad and icotoukinra and how they disrupted space that, again, was traditionally dominated by biologics. I'll talk about suzetrigine, which is a breakthrough in pain treatment. And then talk a little bit about synthetic lethality entries here, Werner helicase inhibitors from Novartis, they're reversible one, and a irreversibly covalent inhibitor here from Vividion. Then we'll talk a little bit about molecular glues and how they've been addressing KRAS. It was a very challenging, one of the big fours in cancer target. We'll talk a little bit about degraders. So we started with inavolisib, which is a monovalent degrader of PI3K alpha. So really remarkable compound. And then we'll talk about the traditional heterobifunctional degraders, KT-474 as an IRAK4 inhibitor, and a degrader outside of oncology, which also is somewhat unusual. And then bexobrutideg to finish it off as a CNS penetrant degrader to finish it off. So let's start out here with laroprostad. Again, I had mentioned how difficult it is to drug PCSK9. And again, it was a target class that's traditionally been dominated by biologics, with Repatha being the monoclonal antibody that targets it clinically. Merck then entered the space with enlicitide, and we were all shocked in 2024 when AstraZeneca disclosed this first clinical oral non-peptide small molecule inhibitor of PCSK9. The whole industry had been trying for a really long time to come up with hits, but most of the even large pharma organizations came up empty-handed. There were some molecules that even made it into development. For example, this PCSK9 inhibitor, or what was thought to be a PCSK9 inhibitor by Pfizer, was even brought into phase 2. And then it was actually found out that it was a translation inhibitor that inhibited the synthesis of PCSK9 and other proteins. And so it was deprioritized and also didn't have the expected efficacy. So for laroprostad, it was really remarkable that this discovery was even disclosed. And what was happening is it did not disrupt the protein-protein interaction between the LDL receptor and PCSK9, but it was binding to a cryptic pocket at the C-terminal domain of PCSK9. And AstraZeneca showed clear crystal structures of that engagement. And again, the proof is always in the clinic. And this compound showed remarkable efficacy, reduced LDL cholesterol here after statin treatment by over 50%. So this is for a patient population that's refractory to statins, and 30% as a monotherapy. And just to benchmark, so first generation statins reduced LDL cholesterol by about 20 to 30%. And then second generation and next generation statins like Lipitor reduced by about 50%. So that's very interesting. And if you then combine laroprostad with statin treatment, you can get to about 80% reduction in LDL cholesterol. So really getting a massive boost over that monotherapy compared to about 70% maximum reduction with enlicitide alone. So again, what this sets up is a nice tableau of a real antibody that's already approved, a macrocyclic peptide that's orally bioavailable but not cell penetrant. So it could have, the metabolism is different, but the tox profile could also be different because we're not hitting any intracellular off targets. And then a traditional small molecule like laroprostad that could have, other safety liabilities, but for example, the cost of goods is much cheaper to produce than Merck. There are not the fasting requirements that there are with peptides. for before the dosing, so there's no like food effect on the dosing. So I think this sets up a wonderful tableau and is something that we very closely watch between those really nicely differentiated molecules, each having a very strong suit. And yeah, it's going to be really interesting to see which one kind of wins out. And I think there's probably space for both. How was laroprostat discovered? Actually, it was from a fragment-based screening. So you see this kind of aminopyrimidine here. That was the original hit. And then with a structure-based drug discovery, understanding that it binds to the C-terminal domain and thereby modulates PCSK9, Dogma Therapeutics and AstraZeneca quickly optimized it to the clinical candidate, solving clearance problems and a number of other problems on the way as well. So more details will be on the Drug Hunter website, of course. The second molecule of the year candidate is icotokimra. Icotokimra is an oral macrocyclic peptide IL-23 receptor modulator. And this comes into, again, a space that's been dominated by biologics. Here, the clinical antibodies, or the approved drug antibodies, I should say, Skyrizi and Stelara, are both among the top 2 best-selling drugs in the US, so making tens of billions of dollars every year. And you know, it's an interesting target space here. We've also seen earlier the entry of deucravacitinib, which is a TYK2 inhibitor downstream of the IL-23 receptor, which provides now an oral option. And that, excuse me. And now we're seeing an entry of a macrocyclic peptide here that shows significant efficacy in a phase 3 clinical trial as a once-a-day oral pill for plaque psoriasis. So again, very remarkable. Nearly half of the patients here actually achieve clear skin in this phase 3 trial after 24 weeks on the treatment. And a couple of months ago, it was even shown by J&J that icotokimra showed superiority over deucravacitinib, which was very, very remarkable. And I think a lot of people got more and more interested in oral peptides. After we're seeing icotokimra, for example, we saw just enlicitide, the GLP-1 receptor agonists that are made available as oral treatments. There's Luna-18, for example, a pan-RAS inhibitor from Chugai that's been very interesting as an intracellular target for macrocyclic peptides. Has recently been discontinued, but still scientifically very interesting case study. And in addition to that, there's for example, from BMS, a PD-1 inhibitor as a macrocyclic peptide that's also orally bioavailable. So really a lot of interest in this particular space, and has been, I think, only furthered by the discovery of icotokimra. Really interesting compound to be watched. And again, a beautiful tableau of antibodies, small molecule drug with deucravacitinib, and a macrocyclic peptide in kind of the same target indication space. So really beautiful to see that so many small molecules can now have like a profound effect on the health of people. The third candidate that I'd love to talk about is suzetrigine. And suzetrigine is an oral allosteric NaV1.8 inhibitor that is invented by Vertex. So this comes as a very interesting entry in the pain space. There haven't been any new mechanisms disclosed in the pain space for over 20 years. And it's been very challenging with some pain treatments like opioids to deal with their side effects. So there's, of course, addiction potential, but also other side effects like constipation, etc. So actually having a new option here in the pain space is remarkable. And suzetrigine got approved earlier this year for moderate to severe acute pain in low oral doses here as well. Remarkably, the compound has comparable efficacy to Vicodin. Vicodin is a combination of opioids and acetaminophen. Again, those are both compound classes that have been studied for hundreds of years. And, NaV1.8 inhibitors are a relatively new class of drugs. Interestingly, suzetrigine has picomolar potency and over 31,000-fold selectivity across sodium channels, which is remarkable given how similar those voltage-gated sodium channels are. And it achieves that by binding to a voltage sensing domain. Here, there's a beautiful pharmacology paper from Vertex on that. Interestingly, NaV 1.8 was not even a target that the industry was very convinced of. It certainly came somewhat out of the blue as a target. And, like NaV 1.7 was much more genetically validated with loss of function mutations known in human, whereas that was not known for NaV 1.8 inhibitors. Yet there have been entries into that field. So Abbott, for example, entered with this furan type molecule here on the left-hand side. That showed really reasonable preclinical efficacy, but was never advanced into clinical development, likely due to issues with PK. So it was very challenging to make this compound orally bioavailable. Pfizer got a step further with this molecule on the right-hand side here, and they were able to actually move towards into phase 2 of clinical development with this compound. Unfortunately, the trial then had to be discontinued for futility. That was somewhat highlights the challenge, in my opinion, how difficult it is to translate in the space of pain medication, because they had seen very good efficacy in a withdrawal model, in a preclinical pain model, but that didn't translate through into this post-surgical dental pain trial. So oftentimes it is very difficult in the pain space to translate your findings from preclinical to clinical. So oftentimes people just use target engagement as their proxy to optimize. And then again, clinical trials in the pain space are extremely painful because, of course, you have a placebo effect. And in that placebo effect, like the pain is very subjective. Sometimes somebody who may feel a 1 in pain for another person that may be a 10. So it's a very difficult space to run clinical trials in. When we looked into Vertex's portfolio, it turned out that suzetrigine was not their first entry into the NaV 1.8 space. Actually, that was VX150. And VX150 is a remarkable compound. It actually got breakthrough designation by the FDA in a phase 2 trial. So it showed beautiful efficacy in that phase 2 trial. However, it was discontinued by Vertex because the doses that were required to achieve efficacy were in the range of 1 to 1.5 grams per day. And that was just not competitive in this kind of space where there are so many different options are available. So Vertex believed that they could get a better compound, and they actually came through with their compound. And that was suzetrigine. So suzetrigine, as you can see, it's a beautiful molecule as well, with 4 contiguous stereo centers. And all of them, and every atom in this, frankly, is required. Vertex tried to really finely optimize this molecule. And even there was a challenge with cost of goods, where the compound was at risk to be discontinued. But then the CMC group at Vertex came through and allowed the synthesis of this compound to be made available much cheaper for a lower cost of goods. And so about 10 years after Vertex had first entered the space of NaV 1.8 inhibition, they came through in January earlier this year with the first approval in this space. So very remarkable story. The next molecule would be HRO 761. That's Novartis's oral allosteric inhibitor of Werner helicase for MSI-high and DNA mismatch repair cancers. Werner helicase is this very interesting synthetic lethality target. It's dispensable in somatic cells, but cells become critically dependent on it if they're basically MSI high or mismatch in DNA repair. So that's a beautiful synthetic lethality setup. The target was discovered by Novartis through an RNA interference screen. And helicases, as we know, are notoriously difficult to drug. So Novartis actually set up a beautiful cascade of approximately like 7 counter screens to come up with just one single hit that they advanced forward. And they basically had a number of nice chameleonic transformations that I want to talk about in the next slide to optimize this compound. which is currently in phase 1 studies, and recent results have been disclosed at the recent ESMO conference. But the optimization is really remarkable. So in those larger molecule space, oral bioavailability often becomes a challenge, and optimizing solubility and permeability at the same time becomes very difficult. So instead of focusing on those macroscopic parameters like, log P or TPSA, what Novartis actually did is they focus on the distribution of conformations in the minimum 3D PSA as a proxy for permeability, and basically a distribution in the minimum log P conformation as a proxy for solubility. And thereby they identified this chloroaniline here on the top as the ideal compound here to go with. And in addition to that, they also had this non-classical zwitterion, which basically can exist in a non-charged state. Let me take a sip. Which, excuse me, which can exist in a non-charged state in the membrane, but in an aqueous environment, it actually gets protonated, and basically in this aqueous phase is much more soluble than it would be in the uncharged stage. So it can behave in different environments. It can behave differently, like a chameleon. So that's super interesting. Another entry, and discovered with a completely different screening method, is this oral allosteric irreversible covalent inhibitor of Werner helicase from Vividion. And this was discovered by a chemoproteomics screen. So, Vividion has this beautiful chemoproteomics platform that is based on Ben Kravat's and other researchers' pioneering work. So what you do there is you basically treat your live cells with covalent compounds, with a library of covalent compounds. Then you treat with a probe, digest with trypsin, and then monitor by LC-MS your signal. And you can essentially see where, like, compared to standard signals disappeared, you can identify like binding sites directly through this assay. So wonderfully, you can identify 3 things through this assay, just in one assay. You can get the potency of a compound, the selectivity across the proteome, and it's also basically giving you a target. So it's somewhat target agnostic that this platform can operate. So interestingly, Vividion discovered this vinyl sulfone compound here, which has a good selectivity across the proteome that they attribute to the poorly or weakly reactive warhead, which is a good thing in a covalent drug. And this compound was advanced by Roche into phase 1 for MSI high cancers, and was recently actually purchased back by the rights to the development were purchased back by Vividion, which is wholly owned by Bayer. So the reason why Werner helicase also is interesting as a target comes from like immunotherapy. So patients that are screened for being eligible for immuno-onc therapy with PD-1 inhibitors are being screened for the MSI status. And there is, of course, a patient population that doesn't respond to those therapies, and a patient population that gets resistant to those. And those are literally waiting patients. So the biomarkers are already characterized. And, a Werner helicase inhibitor could be an extremely interesting option here for them. So how do these molecules bind here? How is the binding different? You can see Vividion's compound binding here predominantly through the covalent interaction with the cysteine, and the rest of the binding is essentially induced fit. And, completely opposed to that, it's kind of this similar kind of binding area. You can see that cysteine here in the back. But Novartis' compound leverages all these polar interaction in this arginine-rich pockets. And, basically, if you look at the D1 and the D2 loop, really obliterates here and opens this loop dramatically. So the binding couldn't be more different in these two here as well. For Werner helicase, there has been a lot of interesting entries. And what is, I think, I learned from this is there's so many different screening assays. We've found like this chemoproteomics platform, high throughput screening. MoMA found a compound by Dell screening. There's so many different opportunities these days for screening methodologies that all can get you to a clinical candidate, which is fascinating. Our next compound here is also a breakthrough. It's a soldonrasib, an oral covalent inhibitor of KRAS G12D on, invented by Revolution Medicine. So this is again a remarkable compound, as KRAS has been long thought of as an undruggable protein. So it's like this essentially marble-like protein. It's a GTPase, and a GTPase, you can't displace GTP. It has femtomolar affinity to GTP, so it's not like in a kinase you can displace ATP. And there were no real binding pockets until Kavan Shokat in this off state found a switch 2 pocket that could be drugged. That were actually, you could drug that, and that's where the first generation KRAS G12C inhibitors, sotorasib and adagrasib, bind to that. But it's actually in the off stage of a state of KRAS. And so essentially what happens there in a mutated form, when KRAS mutates in cancer, it's always on the on switch and very little is in the off state. So essentially only through covalency you can cycle this maybe 1% that's in the off state into that on state and kind of trap KRAS in that way. That's how basically KRAS has initially been drugged. But this is particularly only working on the KRAS G12C mutation, and that's only a minor mutation. KRAS G12D is much, much more abundant. So what if we could drug that KRAS G12D as well as could drug the on state rather than the off state of KRAS? And that's what Revolution Medicine really achieved here. And that's it. I would say it's frankly really in the name. This is somewhat of a revolution of the field, in my opinion. The way how this molecule works is actually through a molecular glue mechanism. You can also appreciate, as the chemist in the audience, essentially, that aspartic acid is much less nucleophilic than cysteine, so it'll be much more challenging to engage that into a covalent interaction. So until very recently, until 2022, it was believed that chemical leverage of aspartic acid to covalently drug KRAS G12D was likely impossible, and that's exactly what RevMed did here. So the way this mechanism works, it's very interesting. The compound initially binds to an abundant cytosolic protein called cyclophilin A. That bicomplex then leverages neoprotein-protein interactions to bind with KRAS. And then essentially in that non-covalent tricomplex, you position that warhead in the right position. And with this particular one, the aspartic acid warhead then snaps in. It's a very interesting design as well. This aziridine, very, very smartly designed. And, you obviously have to make sure that plasma stability is there. So, an incredible job by the team at RevMed here. And if that was not enough, at the same conference, they also disclosed daraxonrasib, which is an oral, now reversible pan-RAS inhibitor that can not only drug different mutations of KRAS, but also NRAS and HRAS. So really a remarkable feat here. And one of the things that people were kind of concerned about then was, well, if this drugs all forms of KRAS, what about wild type KRAS? And wouldn't it be bad to inhibit wild type KRAS? And actually, again, the proof is in the pudding and in the clinic. And essentially what happened here was that there was only a low single-digit amount of grade 3 or higher adverse events in the clinic, showing that this compound is very well tolerated, much, much more tolerated than expected. And there was actually even a complete response in a stage 4 pancreatic cancer patient, an incredibly challenging indication, in a phase 2 trial. And so RevMed is moving this compound into phase 3, and it'll be really interesting to see how this story develops. You can see there's a few changes in the molecule. For example, this thiazole now leveraging the water network within cyclophilin A and KRAS. So really smart design here as well by the RevMed team. And another cool thing about those compounds is that the barrier to resistance may be a little bit higher with those compounds as opposed to sotorasib and adagrasib, where this C mutates or slight mutations in the vicinity happen. Maybe resistance is developing a little bit quicker. But here, as you can see, the binding footprint with those mutational hotspot axis of the compound is very low, so indicating it binds much more to cyclophilin A and lets cyclophilin A do the heavy lifting of the KRAS binding. So even if mutations happen in this particular area, that shouldn't, that shouldn't raise resistance to this compound. So very remarkable compound here as well. Our next compound is an unexpected monovalent degrader, inavolisib. It's an isoform selective mutant PI3K monovalent degrader. So recently received breakthrough therapy designation here and was FDA approved in 2024 for PI3K mutated HR positive and HER2 negative breast cancer. So what is this discovery story of inavolisib, and how was it found to be a degrader as opposed to just an inhibitor? The story is really, really interesting and starts with Genentech's clinical candidate tazelisib. So, tazelisib has been moved into clinical development and actually been discontinued for, there was not enough of an efficacy benefit over existing options. But the biology team at Genentech continued to experiment with tazelisib and found the efficacy in multiple different cell lines could not be explained by the potency or selectivity of this compound. It was continuously more potent than classical PI3K inhibitors like Novartis's alpelisib. So they asked the chemistry team to start a project on this, and maintaining that secret sauce, but not knowing really what this secret sauce was. And the chemistry team, initially was somewhat reluctant and pushed back, but then ultimately agreed to doing this project. And what they found is that during that time is that essentially derivatives of inavolisib degrade the mutant form of PI3K with a 10-fold selectivity window over the wild type form of PI3K, which is very, very, very remarkable, because the problem with general PI3K inhibitors is on-target wild type inhibition that leads to a hyperglycemia effect. For example, here seen with alpelisib, Novartis's compound has approximately 30% of grade 3 or higher hyperglycemia. But yeah, again, developing this compound led to the discovery of inavolisib, which now is selective, isoform selective for the alpha form, as well as selective for the mutant form, leading to a much lower incidence of hyperglycemia in the clinic. Very remarkable story. And I think the mechanism of the degradation is not fully understood. There's some hypotheses out there in the literature as of how it works. But yeah, it's not really fully described. But I think what it shows is if you look for degradation, you'll be able to find much more incidences where degradation plays a role, even when this is a compound where you would look at it as a chemist and you would not obviously identify it as a degrader. Now going to more traditional, and we have about 2 more molecules to go through. More traditional degraders is the compound that I want to discuss here is Kymera's KT-474. It's an oral selective IRAK4 degrader. So IRAK4 has been this very interesting target in immunology, where a lot of pharma companies were working on this, large pharma companies, biotechs, and it was very challenging in phase 2. Efficacy didn't really come through, and people people thought about like maybe the inhibition of the kinase function in IRAK4 to then downstream have an impact on IRF5 and IRF7 signaling may not be enough. So Kymera came in and thought about what kind of advantage can a degrader have here over an inhibitor? So if one would take out IRAK4 as the protein, not only the inhibit the kinase role, it is also part of this midosome scaffolding complex. So that could have an effect if we take out Iraq and that disassembles that midosome complex. That could have an effect on downstream signaling through junk, P38, or even NF-kappa beta, and could have much more durable efficacy. And now this is a degrader actually that's been advanced in an immunology indication. Again, a Serblon targeted degrader. And it's remarkable, in my opinion, because fundamentally all these E3 ligase Serblon binders are derived from thalidomide, and we know the history of thalidomide. So what this, I think, shows is the advancement of safety pharmacology. If you really want to dose this compound to even healthy volunteers, Kymera dosed this compound in 600 milligrams to healthy volunteers, you better be darn sure that it's very selective for only degrading IRAK4 and not the neo-substrates that potentially lead to side effects like Icaros and Alios degradation. Very remarkable work here. Kymera could have salted the earth essentially for everybody who's looking at an entry in degraders in immunology. But luckily, there were no safety, really high-grade safety events with this compound. It was actually recently discontinued for undisclosed reasons, but Kymera is still very much interested in immunology and targeting IRF5, STAT6, and also IRAK4. They have a partner program with Sanofi where they're advancing a compound called KT485 that has an undisclosed structure, but we'll closely monitor that as well. I think it also shows the beautiful differences in PD effect that degraders can obtain over inhibitors. So if you think about classical inhibitors, typically in your PK/PD, you want to make sure that you cover your IC90 at trough, sometimes even like 3 times protein adjusted if you work in virology. But here that doesn't have to be the case with the degrader, and that's beautifully illustrated here. If we look at the single dose of this compound clinically and the percentage of IRAK4 degradation, we see at the low dose of 25 milligrams, we degrade about 30% of IRAK4 after one dose. With a higher dose, 600 milligrams, 300 milligrams, we can get to about 300, about 85, and 600, about 96% degradation of IRAK4. However, with cumulative dosing, even with the low dose of 25 milligrams, now we get to 92% degradation. And that's not something that you can achieve with a simple inhibitor. But here with the degrader, because if you're degrading more of the protein than is newly synthesized, so if you overcome the resynthesis rate, you can have a cumulative effect on basically the degradation, and that can have an effect on the dose, so you can use much lower doses than you would think with inhibitors. Our last compound here, CNS active degraders, and that's bexobrutideg, Nurix's BTK inhibitor here for CML and CLL as well. So what I find really remarkable about this compound is, like, in general, degraders, right? We think about challenges with oral bioavailability because they're all beyond rule of 5 molecules. But certainly a couple of years ago, folks would have laughed at people that claimed that you could get a degrader into the brain, not only orally bioavailable, but also into the brain. But yet this compound here shows a Kpuu in human CSF of 1, meaning that it's equally distributed in plasma and the brain, which is very remarkable. The PD effect is also very remarkable as well, as, for example, in an intracranial tumor model in a mouse, where actually there was a disconnect between the Kpuu. In mouse, the Kpuu was below 0.1, so very low Kpuu, very low brain penetrance. Yet, despite this low brain penetrance, it had an efficacy in this model, showing, again, this unique pharmacology. Interestingly, this compound has in human trials sustained of 90% BTK degradation at 50 to 100 milligrams QD, despite its low bioavailability. Again, this compound has about 5 hydrogen bond donors, and despite that is orally bioavailable and also brain penetrant. And really, the interesting thing about this is one molecule, Nurix recently disclosed this, one molecule of bexobrutideg degrades 10,000 BTK proteins per hour. So it's a catalytic effect this degradation, which is extremely remarkable. Three things I think that I want to cover that differentiate degraders from inhibitors in pharmacology is like one, for pharmacology of inhibitors. Again, we said potentially if you cover the IC20 the first day, you're not going to cover the IC90 the next couple of days. And with a degrader, as we've demonstrated before, you can have that cumulative effect. And also, if you think about the dosing of an inhibitor, typically at the end of the dose, you want to still cover C trough. But with the degrader, even if you degrade, like all of the BTK protein in the first 2 hours, and it doesn't come back for 12 to 24 hours, then you still, you don't need to cover your C trough at the end of the dosing day, and that could lead to a much lower dose. And again, also that cumulatively over the lifetime of the dosing, you also don't need to cover that, EC90 or IC90, because you can essentially degrade as long as you overcome the resynthesis rate of the protein. You'll be essentially fine. And that can help with if you have like CYP expression or if you have mutations that make your compound less effective. With those, essentially, degraders could still be effective if they still overcome the resynthesis rate. And just to show that this is not a unique finding for bexobrutideg, there are other degraders now as well, more and more that are emerging. Here, one from Arvinas, that is a LRRK2 degrader. We're recently in the patent literature for this particular compound. They have shown in multiple compartments of the brain, the cortex, the striatum, the cerebellum, that this compound can, deeply degrade LRRK. So again, showing that, CNS active degraders are possible. Again, it was very difficult, I guess, to do justice to all these case studies in just 1 to 2 slides per case studies. We raced through all of these here today. So if you're interested in learning more about those compounds, all of this will be available for you on the Drug Hunter website. And here I want to, of course, call for your nominations of compounds that you think made a significant impact in 2025. So we're really looking forward to your nominations. Feel free to email them to info@drughunter.com. We also will be sending out a follow-up email where you can then essentially nominate your molecules as well. I get the privilege to talk about all these amazing stories here, but of course, the researchers in the field get the credit for discovering them, and our team gets the credit for summarizing all of this research that I have the privilege here to present. With that, we want to acknowledge everybody who contributed. And of course, I'd love to answer any questions that you may have. And with that, we'll stop sharing here. Okay, let's go. I'm going to open the Q&A box. Thank you so much. We have a question from Yoshi. In recent years, the protein targets are becoming arguably more complex and have less obvious druggable pockets. Although it could be hard to define a drug compound's complexity, do you think compounds are becoming more complex in recent years to address the more complex protein targets? Excellent question, Yoshi. I think you're right. I mean, people are addressing those more complex targets with more complex compounds, but I wouldn't say like you have to go into proteins immediately or like macrocyclic peptides or degraders. I think for every target, there's the right modality, and clever design oftentimes can make multiple compounds possible. So, for example, if we take that Werner story with Novartis's compound that looks somewhat complex as a reversible inhibitor of Werner helicase, but had very smart design choices. Then you have that Vividion compound that looked very simple, yet a lot of thought went into that. A different screening method went into that. The target was identified somewhat differently. So you don't need necessarily that complexity in the chemical space matching the complexity of the target, but it's the smart design, the smart choices of assays that I think are very important. Great question. Next question was, or do you think compounds will be increasingly more complex in the future to achieve novelty, both from a patent perspective and from a marketing perspective? Yeah, that's a great question. I think from a patenting perspective, we're seeing much more activity on like Me Too compounds. I think one of the things with the advent of AI, we're seeing much more activity in the space of Me Too compounds. So yeah, increasingly, I think yeah, there's coverage there. But I think the what I believe is that there's so many, like, again, new opportunities in small molecule drug discovery, that there's a lot of space for a lot of new innovation in many different fields. One of the fields we didn't get to talk about, for example, today is like the whole field of obesity and GLP-1 receptor agonists. There's like huge activity going on there with orforglipron being a very exciting compound. But then there's follow-up compounds. There's combination therapies that could have different impact. So yeah, a lot of what I would say is a lot of different opportunities. I haven't seen a more exciting time in small molecule drug discovery. Thanks for the question, Yoshi. We have another question. That CSF KPUU is not the same as brain KPUU for bexobrutideg. CSF blood ratio is easier to permeate. Don't call it brain penetrant. The evidence is lacking. Okay, yeah, that makes sense. So there is actually it is pretty interesting for bexobrutideg. Again, they've shown human CSF data, of course, but they have actually also shown preclinical data in different species. Interestingly, in the mouse, there is somewhat of a disconnect. So in other species like rat, the KPUU is more towards 0.3. They've also looked at the KPUU in cyno, and it's much more closer to the 1. And that's an actual KPUU where they did homogenate the brain and not only look at the CSF fluid like they did in human trials, what you would do in human trials. So yeah, I think it's brain active. We can agree on in preclinical species it's brain active, where essentially we've seen efficacy in preclinical mouse models. It's very, yeah, I will agree. It's very challenging to believe that this is a brain penetrant compound. It also is actually effluxed. I recently looked at a poster, and it has an efflux ratio of about, I think, 20. So it's not really traditionally as compound that you would think is brain penetrant. The hydrogen bond donors, the 5 that I mentioned, multiple of those are actually masked internally, which I think contributes to potentially the compound at least getting into the brain. And again, it has catalytic activity. So one molecule of bexobrutideg degrades 10,000 proteins of BTK. So even if one molecule gets into the brain, it still degrades 10,000 of those proteins. It's a great question. I definitely understand. I have my challenges with this molecule as well. Thanks for the question. Sina is asking, could you please explain again why Werner helicase inhibition attracts this much attention in drug discovery? Yeah, that's a great question, Sina. So I think it's the one reason that I think it is so attractive is if you look at immuno-oncology, right? People that are being screened for eligibility to PD-1 treatment, so a pembrolizumab treatment, for example, with Merck's PD-1 inhibitor, their MSI status is already checked, right? So you have a huge patient population that has already a characterized biomarker, and not everybody who receives PD-1 therapy responds to those. Yeah? I think the response rate is approximately like maybe 20 or so percent. And for the people that are responding to the therapy, there's development of resistance on top of that as well. And for those patients, at least Werner helicase inhibition is an option as a potential combination partner. But what there is there's a large patient population that already has the MSI status characterized as a biomarker, which is why this is so attractive. Also, the synthetic lethality is attractive in my opinion. So you can get a much larger therapeutic window with the synthetic lethality target, of course, where essentially you're only targeting those MSI high or DNA mismatch repair cells and leaving those somatic cells somewhat untouched because Werner helicase, again, is dispensable in those somatic cells. So yeah, it is an attractive target. Again, clinical data has to come through. Recently, again, Vividion and Novartis disclosed clinical data on their assets. And there are kind of a few questions with the overall response rate. But again, it's an interesting target to be monitored and yeah, to be definitely to be watched. Okay. Yoshi is asking, I've heard you and Lou say that they're excited about the future of small molecule drug discovery. Is this because new modalities are sparking creativity, or is that because of the new organic chemistry methods facilitating the synthesis of previously unseen structures? Great question, Yoshi. We haven't talked about the synthesis yet, but yeah, you are absolutely right. I think it's both. It's not one or the other. If you look at this Merck molecule that I showed in licitide, it wouldn't have been possible with traditional synthetic methods. A lot of biocatalysis actually came to the rescue to make this molecule. I think in the macrocyclic peptide space and in the peptide space in general, we're seeing much, much more biocatalysis now, which is very exciting and opens up a space that hasn't been accessible there, synthetically before. And the same, I guess, the same is true for, like peptide stapling methods for, yeah, generally, modern organic chemistry, but also the target classes that can now be addressed. Again, there were a number of target classes that we walked through today that were traditionally dominated by biologics, where now we're seeing entries of small molecules that can drug these targets that previously were thought undruggable. IL-17 is another one we haven't been talking about. That's interesting. Again, the GLP-1 receptor agonist space and GIPR and all these new targets for obesity will also be really interesting to watch. And yeah, in addition to that, the whole pain targets that we just briefly only touched upon with suzetrigine. There's a number of other interesting targets and compounds that are just really interesting to see come through. And I think small molecules can achieve, like, they're oral, right? They're simple to take. Essentially, it's low burden for patients. So yeah, they have unique advantages that other modalities don't have. And then the whole exploding induced proximity field that we kind of started to cover with monovalent degraders, right? We talked about inavolisib a little bit, but there's so much more going on in this induced proximity field that we haven't had the chance to touch on today. But yeah, I think there's an incredible amount of stuff to be excited about. Yeah, and somebody would have nominated Pfizer's atirmociclib, the selective CDK4 inhibitor, as the molecule of the year for breast cancer. That's a great nomination, actually. Thank you for that. Yeah, CDK is also another interesting field. Of course, CDK4, CDK6 inhibitors are somewhat standard of care in combination for HR positive, HER2 negative breast cancer, but it's been difficult to dial out that CDK6 activity that may be responsible for some of the off-target effects. So the breakthroughs that Pfizer is showing with more selective CDK4 inhibition and also others with selective CDK2 inhibition, I think there's huge excitement there, because actually one of the things that happens with dual CDK4, CDK6 inhibitors is the development of resistance on therapy. And it's been shown that CDK2 inhibitors can rescue CDK4, CDK6 resistant tumors to be sensitive again. So yeah, really remarkable discoveries there in this field as well that we didn't have time to cover. But yeah, thank you for that. And with that, if I don't see any more questions, we're slightly over time. So I want to thank everybody for attending. Thank you for your engagement and for the questions. We really appreciate that. We wish everybody a wonderful holiday season and a wonderful rest of the year with your loved ones. And then we will be back here next year for you, and hopefully, start the year with a firework as well. So stay tuned for what's to come in the new year. And again, thank you everybody for attending. And with that, I wish everybody a wonderful rest of their week, and hope to see you soon.